Hi, so my name is Katie Hughes. I am the manager of informatics at Inzen Therapeutics. Today, I'm going to be talking about joining LabKey with external third-party data warehouses, namely Benchling, and how we can use those data warehouses to inform our development of our various pipelines that we have here. First, an outline. I'm going to be talking about a brief introduction to Inzen Therapeutics and the concept called Thanokine biology. We'll be integrating bench scientist tools, Benchling with LabKey, and I will be giving you an example workflow, which is our proteomics pipeline. So I'd like to introduce a concept called Thanokine biology. And living cells are constantly processing their environments, and other living cells can provide signals which inform these cells about health and disease. An example of those could be cytokines, neurotransmitters, hormones. Living cells can actually also process information from dying cells, and scientists here and elsewhere have found that the same cell can die in various different ways, and that these signals that it releases can vary based on how this cell is dying. We've coined a term thanokine to mean a signal that a dying cell releases. Thanokines can provide insight into the health and disease of the dying cell, and our hopes are to capture and analyze these thanokines for therapeutic potential. So in summary, thanokine biology is the study of signals that a cell releases during cell death process. Now I'd like to talk a little bit about our rationale in integrating LabKey with Benchling. Our goal in the data science team is to answer questions for bench scientists quickly. We would like to be able to ask questions like, which thanokines were detected in my specific experiment? So this is a very specific question. Maybe a scientist would like to go a little broader and say what common thanokines were shown in a certain cell death type. And we've also answered questions that were very broad, such as, are there thanokines that are similar across all experiments? And in the informatics group, I have two pieces of philosophy. Number one is meet bench scientists where they are, and number two is use the tools that they use. For Inzen, one of those tools is Benchling. It has an electronic lab notebook, inventory tracker, and sample registration, and our bench scientists are pretty integrated in this tool. Additionally, it has a warehouse for storage, and that this will become relevant later, is that this warehouse is in Postgres. Our major challenge here is that the data and metadata live in two distinct warehouses. In Benchling, we have the metadata for a particular sample. So it might contain what cell line this is, what media was used, and the mode of death. In LabKey, however, this is what our data science team uses almost exclusively. And this contains all of the omics data that we have generated, metadata on what's measured, hit lists from previous experiments, and curated annotation sets. And so, as I talked about before, our lab scientists use Benchling, ELN, inventory, but the data scientists are more like to use LabKey because it's a more flexible data warehouse. So how do we keep this as a single source of truth? And our solution to this would be to expose the Benchling warehouse in LabKey. So we want to keep LabKey as a single source of truth. Bench scientists use Benchling. So how do we efficiently expose this in LabKey? And so now I'd like to go through some technical details on implementation and examples. So for those of you who are familiar with the LabKey data warehouse, you may be familiar with some of the things on this slide, this query schema browser. And the query schema browser, contains all of these schemas that are located here. But when you expose a third-party warehouse in LabKey, you get an extra little schema table over here. And this is something that your LabKey personnel manager can help you to expose. And so here we have our Benchling warehouse, and we have two different kind of pieces of things that are exposed here. One of which is the biological metadata. That's stuff that's input by the scientist. And then our technical metadata, this is information that's input by our proteomics team, which I'll talk a little bit about later. So what are we actually doing here in terms of our thanokine? So we're trying to capture these thanokines through proteomics, and if the same cell can die in different ways, we'd like to be able to capture which different thanokines are released in different cell death types. This could give us insight into the health and disease of the dying cell, and our goal is to store these into a database, probably LabKey. So I'm going to go through our integration pipeline here. So our lab team will input the biological metadata into Benchling, and then the proteomics team will put in the technical metadata into Benchling. And then the next part is our bioinformatics team thinks about the analysis and the analysis metadata that we're collecting. And what we're going to do with that is we're going to check that against an exposed Benchling warehouse in LabKey. Use some of those, our LabKey API. And I'll go into more details here. There is a red circle around this because this is a very important step. And this ensures that we're not inputting data or metadata that might not be correct into LabKey. If the validation check passes, then the analysis pipeline continues. And finally, we commit our results to LabKey. Now, this is a commit very similar to what you would think of as a Git commit. So we can revert our commits, but the data should be correct based on our validation checks. But we consider this very similar to a Git commit. So what we're going to want to do now is we're going to want to create an R object from this Benchling schema. And to maybe walk you through some of this pseudocode is, so we'll basically join all of those Benchling tables that we were able to see within that Postgres schema that was exposed in LabKey. So we're going to select all of them and join all of them. And then we will find some individual criteria, maybe some kind of sample ID or something of that nature. And then what we're going to do is we're going to call this execute SQL from the LabKey API. And here is where we'll enter our Inzen TX server. Here's where we'll enter our specific folder path. So we have a folder called external data sources. And then finally, we'll enter our specific schema name, which we will call Benchling in LabKey. And so now what's returned out of this is an R data frame that has that is, what's returned from this is a R object that is from this exposed benchling schema. So our next step is our validation checks. And as I said before, these validation checks are incredibly important. And what the input is here is the bioinformatics sample sheet. So this is all the analysis metadata that we are talking about prior. And additionally, we will put in the Benchling warehouse and LabKey, our object that we just created. And from here, we will have, we'll basically filter on a criteria that might be in a column or something like that. And we'll do that for both this bioinformatics sample sheet and also the Benchling warehouse. And then we check to see if all of that criteria passes. And if it doesn't pass, then an error is thrown. And if it does pass, then it becomes validated. And what we create here is an enriched set of metadata that's carried along through this data pipeline. And so that's what we return, this enhanced bioinformatics sample sheet. So let me just remind you of our goal. Our goal is to answer questions for bench scientists quickly, and they could be something, you know, are there thanokines that are similar across all experiments, which is very broad, less broad, which would be what common thanokines were shown in certain cell death types, and finally, very specific questions like which thanokines were detected in my specific experiment. And where that comes from and where we're able to answer these questions is through this commit results to LabKey set of functions. And so after the analysis pipeline is run, these results are committed to LabKey. All of the omics data raw and analyzed, a hit list from these experiments, upregulated and downregulated. And loading these into LabKey provides us with almost a continuous integration of results where we'll find hit lists from previous experiments, curated annotation sets, and from here we can run, you know, very quick GSEA type analyses on some of these previous experiments. And yeah, that is our pipeline in a nutshell. And some of the cross-tool integration is going to be a necessary part of many pipelines to come. This is the future. And so what this does though is it minimizes your credential input. So you'll only have to put in one API key versus many API keys. LabKey has been for us and can be for you one-stop shopping for, you know, Benchling, even Postgres or SQL databases that you may have externally. And here at Inzen, we've developed our core analytical pipelines around LabKey to provide continuous integration of experiments so that every time we have a new experiment, our data grows and we're able to check against that. And this has not been a one-person team. This is a large team that I work with of wonderful people, and I would like to thank them all. And additionally, we have some great help from the LabKey team. Steve, Bernie, Hannah, John, others have helped us a lot. So thank you very much.